Hello everyone, today we will talk about the Coombs test. Coombs test is also known as antiglobulin test. So here in Coombs test we will study about its definition, then anti-RBC antibodies, what is anti-RBC antibodies called here, and then we will study about the Coombs reagent, what is Coombs reagent and when and why we use Coombs reagent, and what is direct Coombs reagent and what is indirect Coombs reagent. Now starting with definition, antiglobulin test. What is antiglobulin test? Antigen and antibody reaction. When an antigen and antibody react, we know in the RBC area we have antigens and there are many antigens. As I have taught you in previous class also, there are many blood group system. It doesn't have only one blood group system, they are more than one blood group system, and more than one blood group system means we have many antigens also. So here we will study about how an antigen react with antibodies, an antigen and antibody reaction test, and this reaction test is used to detect the presence of anti-RBC antibodies. What do you mean by anti-RBC antibody? Means the antibodies which is against the RBC. Okay, the antibodies which kills the RBC, which is against the RBC, is known as which is harmful to that particular RBC, is known as anti-RBC antibodies. So in this test, antigen and antibody reacts in the presence of Coombs reagent, and this reaction is used to detect the. So as I was telling that in Coombs test, antigen and antibody react, and this react in the presence of Coombs reagent, and this helps to detect the presence of anti-RBC antibodies. Okay, now these anti-RBC antibodies are of two types, complete and incomplete. So in complete, it comprises of IgM, a type of IgG, a type of antibodies. Incomplete, and in incomplete it comprises of IgM type of antibodies. Why IgG as complete antibodies and why IgM as incomplete bodies, I will explain. So as I have teach earlier that in blood groups, in RBC there are many antigens. So suppose we consider ABO blood group. Okay, so in ABO blood group, A blood group will have antigen A and antibody B. Blood group B will have antigen B and antibody A, whereas blood group AB will have antigen AB and no antibodies, and blood group O will not have antigen, it only have antibody. Okay, that is A and B. Now we have antigens. In the RBCs we have antigens. Now these antigens, as I told earlier, the antigens of blood group system ABO is oligosaccharide. Okay, it's a carbohydrate, it's an oligosaccharide. We can call it as a sugar candy. Okay, so it's an oligosaccharide. It's a carbohydrates. The antigens are carbohydrates. Whereas when we talk about IgM type, in IgM type, RH blood group system, in RH blood group system, the antigen D is present. The RH antigen D, the RH antigen D is protein. Okay, you can call it as chicken piece, like chicken have a protein. So the RHD antigen is a protein which is IgM type, and blood group ABO, their antigens are basically carbohydrates. Okay, that is oligosaccharides. So it comes under IgG. Now the IgG blood group system here, when there is an antigen antibody reaction, when IgG antibody is react with the antigen, it agglutinate. Okay, whereas when IgM react with antigen, the IgM antibody when react with antigen, it does not agglutinate. Okay, so we can again comprise it as a agglutinate antibodies and non-agglutinate antibodies. Now, why IgG is the agglutinate type of antibodies and why IgM is non-agglutinate type? IgG is basically pentamer. Okay, if you remember the antibody structure, it has two light chain, heavy chain, one at bond with disulfide bond. It has two FR region, there is a bonding region, and one FC, that is a receptor region. Okay, now so basically in IgG type it is a pentamer. Pentamer means it has 10, like two binding side, so 5 into 10 means 10 binding side, and these it has 10 binding side, whereas in IgG, as I was telling, that IgG is a pentamer, therefore it has a 10 binding side, that is called as decavalent. Okay, and its FC centers are joined. Okay, now whereas talking about IgM, it is a small antibodies and it has only two binding site and one receptor site, the FC center. Okay, now it is a small. When there is a RBC has a negative charge, so more than one RBC it will has a tendency to repels, as we know like charges repels, and this repulsion property is known as zeta potential. Now suppose pentamers, it has a 5 binding site. If two binding site is attached with one RBC, other two binding site with other, now it has within other two binding site with attached with one another RBC, and it has a tendency to cross link with each other. Okay, whereas in IgM type, it is a small antibody, and as we know RBC is a negative potential, it has a negative charge, it will repel, and one antibody, it has only two binding site, one antibody attached to one region of RBC, another in another RBC, and another one in other RBC, and as it has a negative charge, it will repel. So there is no property of cross linking with each other. As it doesn't cross link with each other, therefore no agglutination is formed. Whereas in IgG type, there is a cross linking which forms agglutination. Therefore it is called, as one is called agglutinate type of antibodies and other is called agglutinate type of antibodies. Whereas in the case of IgG, it already agglutinate. We do not require Coombs reagent. Why? Because like in Coombs reagent, when added in IgM type of antibodies, and when Coombs reagent is added, it will bind with and it will form antigen antibody reaction. It will form cross linking and it will help to detect the antibody, anti-RBC antibodies. Okay, so in case of IgM type of antibody, we use Coombs reagent. Clear? So end of the topic. Will